Good morning, dear students, and welcome back to our chapter number 2.3 viruses. Okay, today we are going to start with bacterial virus, or we can say bacteriophage. So, the virus which attack bacteria, okay, the virus which attack bacteria and multiply within them are called bacteriophages. So it parasites on bacteria. Bacteriophages were discovered by British scientist F.W. Twister in 1915. Detailed study of the bacteriophage were made by the French scientist D. Herrle in 1917. D. Herrle observed that some invisible agent was destroying the culture of dysentery bellicle on culture plates. So he also observed that this is a special type of virus and was obligated parasite on E. coli, which is found in the abundance. So in the intestine of human being and other mammals also. So the bacteriophage that attacked E. coli, that is called coliphage. ठीक है, coilphage. So the structure or appearance of the bacteriophage is like tadpole or frog. So its body may be divided into following parts like head, neck, tail, basal plate or tail fiber. You can see here, this is the structure of bacteriophage. It is divided into many parts. See here, this is the structure: head, neck, tail, basal part and tail fiber. Clear? So basically bacteriophage structures is look like that. So this is the outer line, and in the B point we having electron microscopic structure of bacteriophage. So DNA as nucleic acid is found in the central part of the head. It is surrounded by protein coat, and the DNA molecules is double stranded and linear. Okay, that means the tail is the tubular structure covered with the contractile sheath. Can you see this? Yes. Now neck or collar. Okay, neck and collar is, when neck is present, that means collar is also present. So at the junction of the head and tail, at the basal part of the tail, there is a tail plate. So it contains 6 thin tail fibers. Okay, and tail fibers perform two functions, that is attachment with the host surface and secretion of enzyme from lysis of host cell wall. Clear? Now, reproduction in bacteriophage, or we can say the life cycle of multiplication of bacteriophage. So, bacteriophage themselves cannot reproduce. Thus, for reproduction, they attack bacteria which act as host for them, and the steps of the reproduction, let's see. First is attachment. Okay, so they get attached with a protein of capsid of the virus attached on the host surface. Okay, then penetration. So in this, DNA of the bacteriophage enters into the host living protein part outside. Clear? Now next we having is biochemical replication. In this, DNA of the virus replicate in bacteria body and produce large number of DNA. Okay, and next we having virus assembly. So virus assembly means when DNA covers themselves by using protein of host to form complete bacteriophages virus body. Clear? Now at the last they release. So virus give pressures to the bacterial wall, thus the cell body of the bacteria burst and bacteriophage virus are released. Okay, so I'll show you the cycle how it looks like. You can see this is the life cycle of bacteriophage or reproduction in bacteria. Clear? Yes. So now next we having is transmission of viruses. Okay, how they get transmit. So the virus are transmitted from one organisms to another by following ways. So first we having by insect vectors. So the insect which carry the virus from one host to another host, that is called vectors. Okay, like leaf hoppers, white flies, flea beetles, they sucks the sap of the diseased plant so that virus reaches to their stomach and finally to the salivary gland. When such insect attack other healthy plant and sucks, so what it happens, they snaps, okay, they saps, and the virus are transmitted into them through saliva. Clear? So next we having by seed also. By seeds also they can attack. So the seed of the dispersal plant carry the virus particles, and these seeds are shown in the field. The young plant get infected. Okay, so seed transmission of viral is not common. Now next by contact. Okay, so virus can be transmitted mostly by contact or slightly rubbing of the infected and healthy plant organ, that is called transmission of TMV. Clear? Now next we having is by pollen grain. So the pollen grain of infected plant infect the healthy plant after pollination. Example, for example, we having bean mosaica virus. Okay, it also transferred by root. So by root of infected plant are also responsible for transmission of virus. When root of the infected plant come into contact with the root of healthy plant, they infect. And example we having yellow mosaica virus of wheat. Clear? So in the same way, by wind and water, by weeds, by nematodes, and by mechanical means also they get attached or transmit. Okay, so let us talk about the economic importance of virus. So useful aspects. Okay, so as in the form of link between living and non-living, does it help us to understand organic evaluation? Okay, now we can also do the biological controls, like bacteriophage eat bacteria, therefore bacteriophage are used as biocides to the destroy of many harmful bacteria that present in the polluted area and can also used as a bioweapon. Okay, for example, water of river Ganga always remain pure and clean, so in the bottles year after year due to presence of bacteriophage in it. Now they also having harmful aspects, so like human disease, animal disease, and plant disease that we have already discussed, right? Now let us talk about the difference between bacteria and virus. So here is a table given difference between, you can see from here, right? Now my dear students, next we having is viroids. What is viroids? Virus, see this, है ना? Viroids means, virus means poison, aeroids means denutritive. Okay. So denutritive, that means viroids are smallest self-replicating plant pathogens which were discovered by Denier in the year 1971. It is the smallest single-strand inflected RNA particle, okay, that do not contain protein code, that is capsid, and it composed of RNA having 300 to 400 nucleotides. So let's see what are the features. See, this is the figure of a viroid, that is PSTVD. Okay, so let's see the features. So viroids are smallest infectious particles of RNA, similarly to RNA virus but lack of capsid. It has low molecular weight. It can multiply by both RNA dependent and DNA dependent replication. Clear? Now let's see the difference between virus and viroids. You can see by this table. So here your chapter is completed. I hope you have understood. Yes. Thank you.